in recent years, quantitative treatment of the aging process all but transformed the field. I mean, the field really grows exponentially in terms of the contributions, papers, and discussions, and featured at conferences and panel discussion like this one. However, there is still a lot of skepticism in the field. Some scientists simply don't accept the clocks and the use of biomarkers and kind of rely more on, for example, hallmarks of aging or other features of the aging process. So today in the panel discussion, we are going to figure out what is the applicability of the clock, the actual applicability of the clock, what are the best clocks, and we will hear from the best experts in the field, I think. So our panel participants are Kristen Fortney, she is the CEO of company BioAge, Peter Fedichev, he's CEO of company GERO, Steve Horvath, he's a professor from UCLA and a pioneer in developing the aging clocks, and Stanislav Skakun, who is the CEO and founder of the Biodata platform. So welcome everybody. And what I plan to do is that I will be asking some questions, and hopefully you can answer them. And I suggest that we do this in a way so that it's clear to not just expert scientists who work in the field, but who just regular people who would like to learn about the actual use of the clocks for particular people, for example, in the clinic, but as well as in generally like in fundamental science. So my first question is that many clocks and biomarkers have been developed, as you know, but it's kind of confusing now in the field because there are just too many. It's impossible even to read all the papers. And of course, the presentations feature mostly good examples of the use of the clocks. So could you maybe just summarize, in your opinion, what do you think are the best actionable clock? What is the actual best use of the clocks based on your experience? And I don't know, maybe Kristen, would you like to start?
Yeah, sure. So we've looked at clocks in a number of domains. There's methylomics, proteomics, metabolomics. My personal favorite type of clock is a clock that doesn't just tracks your age, but also predicts the future. Because of course we already have a variable which tracks your age very well. It's called your age. And the more potent biomarkers will also predict, your future health, your future longevity. So we're very interested in those clocks that predict all-cause mortality. Ideally, long-range all-cause mortality. That's another important point, because if you have a very elderly cohort of people, it's pretty easy to predict, who's going to die in 1 to 2 years because there's so many different things going wrong. But long-range clocks that predict, a couple decades out, I think are really capturing what we care about when we're talking about aging. And I think that there's a few different modalities that work. I've seen good publications here, and in our own data sets as well, in methylomics and in proteomics and metabolomics. And I'm excited to sort of evaluate these in the clinical setting, hopefully as early as next year. Because again, the mark of a true clock is whether it can capture the extent to which an intervention is rejuvenating an older person.
Okay, thank you very much. Yeah. I don't know, maybe Steve, are you there? Could you maybe continue? Steve,
Can you hear me now?
Yes.
Yeah. Okay. Coming to the topic that many publications are coming out, and they describe very exciting clocks in many areas. We mentioned proteomics, metabolomics, any omics, transcriptomics, and of course clinical biomarkers. Above all, I need to remind everybody that there is a crisis of replication in the entire biomedical research arena. It's not just biomarkers, and there are these scary papers where people claim half of all the articles report false positives, and we could have a 1-hour discussion. Is it 50% of articles or is it 80% of articles that contain exaggerated claims? Hopefully it's less, but the bottom line is, before any of the biomarkers can be used in a human clinical trial, we really need to replicate them, and in many different ways replicate them. So above all, claims have to be validated by several independent groups, several independent cohorts, before we can even take them seriously. That's one comment. The second comment is, of course, we want to have as many biomarkers as we can. So we mentioned hallmarks of aging. Ideally, from each hallmark of aging, we have 1 or 2 or 3 different biomarkers that represent it. But this is an ambition. Then there's a reality. So again, it really is up to the people who work in these different arenas to develop biomarkers that can be replicated. Hopefully we will have many, because there are 2 ways to look at the literature. One is we have way too many clocks, hundreds of clocks have been published. But the other way to look at the literature is it's very disappointing how many biomarkers are truly trustworthy in the sense that they have been validated in dozens of different populations. And now, it's the best validated biomarkers are, of course, those that are already being used in the clinic. C-reactive protein, albumin levels, various markers of kidney function, liver function, and pathology, biomarkers that are being used. Now, they clearly predict morbidity, pathology, but very much what Dr. Fortney earlier said. These biomarkers may be terrible at predicting bad outcomes in the decades away, because in my worldview, it is attractive to have a biomarker that even applies to a 30-year-old. Why? Because we believe that aging starts relatively early, which is another topic. When does aging start? But I think nobody will dispute that aging is already manifest in a 30-year-old, no question about it. And so ideally, a biomarker applies to the entire age range, from early adults to a 90-year-old. And also, it would be attractive to have a biomarker of aging that applies to many different populations. I hope we don't need to distinguish aging in different populations. I want a single clock that works in Asians and also in the European ancestry and many other ancestries. Ideally, we have biomarkers that cross species barriers, right? Why? Because people use rodent models, and it would be nice that that you have a biomarker that applies both to rodents and to primates. And when I survey this field, there are very few biomarkers that satisfy these criteria that I mentioned. And there's a reason why I love methylation, because I described already biomarkers that apply to rodents and primates at the same time. These biomarkers have been validated really in virtually all populations. I always tell people, I give them a money-back guarantee that they can validate GrimAge when it comes to predicting mortality risk. I've never seen an epidemiological study that did not validate GrimAge as mortality predictor. So I'll stop with that. Yeah.
Thank you, Steve. Peter, would you like to continue?
Yes, thank you. Well, I think that everyone wants to have biomarkers which do predict mortality. Or if you're in a biotech, maybe the long range is not the most important thing for you, but nevertheless, you want to know what's going on and if these biomarkers capture the effects of drugs. From the fundamental science point of view, I would like to stress here is that one way to develop these biomarkers is to explain the data. Everyone wants to have a good predictive biomarker. But I think not the less important issue is to find biomarkers which also explain the process. So we need to get understanding, not only to get the exact number, but we want to actually understand what's going on. So for example, I will give you just one example. We know that clocks which we are using are associated with chronic diseases, which are very difficult to reverse. So the effect of chronic diseases obviously sticks for a very long time. But also these clocks respond to lifestyles like diets and smoking. And we know that if you are not yet sick with a chronic disease, then the effect of smoking on biomarkers is totally reversible. So it clearly gives you an idea that there are two kinds of processes involved there. And a single number like a clock captures a combination of the two. So I think that in the future, the accuracy is one thing. Of course, black box clocks will work and will give us good clinical tools. But on top of that, just to distinguish between slowing down aging process and rejuvenation, we will need to find out which components are actually contributing to that single number. Basically, I'm calling to understand how many clocks are actually there. Mortality clocks, chronological age clocks, what is the minimum number of clocks with different properties, and not just different source of signals are there, and what kind of biology is behind that. So I think these are the most maybe interesting and challenging issues ahead.
Thank you, thank you. And Stanislav, what are your thoughts on this question?
Yes, thanks. Well, I think one of the most important things and mysteries about aging is that a very multifaceted process which runs unevenly in different systems and organs of the organism. And therefore, what Steve is saying and what Peter is saying is very important, that we are going to have a lot of biomarkers of aging. We will need to have them stand as close as it's possible to the aging process itself. Therefore, so that we can be able to check the causes of aging, which are not yet fully clear to us. And therefore, we will be able to track different velocity of aging, different speed of aging in different systems. So the more comprehensive the clock is, the better. The clock may not be simple, I think.
Thank you. I'd like to actually clarify this first question, because I really want the listeners to have a very concrete recommendation. Let's say somebody works on mice and they want to test the effect of a particular intervention that they give in the diet, or maybe they want to know their biological age. So there's a lot of companies around there, and a lot of people and organizations claim that they are able to do it. So what is your advice, both from the mental scientists and in the practical use of the biomarkers and clocks? I mean, the question is to everybody. Yeah.
May I jump in?
Sure, start. Yeah.
I will explain why. Because I'm tracking several hundred biomarkers every month of myself. Therefore, I'm having a hard job interpreting this and trying to determine a biological age each month. So I think that the advice that Steve gave, that the clinical biomarkers will be at the moment the most precise for tracking your health condition, is correct. The thing is that these biomarkers are the most easily to obtain. They have been mentioned: the C-reactive protein, albumin, transaminases, I mean liver and kidney markers, and some other biomarkers which have wide clinical application. But the issue here is that sometimes one measurement is not enough. So you should have a very longitudinal, long data of yourself. You should measure them regularly to avoid oscillation, to avoid the chaotic movement of the biomarkers, which may occur when you have a cold, when you have some health condition, for example, and when it's gone, the clinical biomarkers, they jump back. So the best idea right now is to collect some basic biomarkers, but to do this regularly, so that you can determine what's your personal individual norm, what your health usually looks like, and to aggregate the data. So I think that the clinical biomarkers would be the best option for a regular person, a non-scientist, to use right now. Thanks.
Thank you. Who would like to continue?
Okay, I think that's a great question, Vadim. It's a super practical question, right? And it's one that actually my company is confronting now, because we're running our first clinical trial next year. And I want to know if this therapy is also going to rejuvenate people, and what biomarker do we use? So my simple answer to the question is that you don't have to choose, right? Why choose when you don't have to? a lot of these omics modalities, be it proteomics or metabolomics or methylomics, right? You can just, with modern technologies, spend a few $100s and measure millions of different sites if you're looking at the methylome. And then that'll be compatible with, you can evaluate, several dozens of these biomarkers, which, there isn't really a one true winner yet. But by going to omics profiling, you can keep your optionality open. And I agree with Steve that, the methylome is the best validated so far. It's just been done in so many different cohorts. So that's like a clear decision to make, where you can always go back in time and reevaluate as the field evolves.
Thank you. And Peter and Steve, I don't know who would like to continue.
Yeah, I mean, when it comes to mouse studies, or more generally anti-aging interventions, let me start with the following. With a trivial statement, aging is very complex, right? Many pathways are involved, many different pathologies. And it's hard to imagine that there is one biomarker that captures the beneficial effect of any good treatment. I give you an example. Blood pressure medication, right, is a phenomenal quote-unquote anti-aging intervention. You take your blood pressure pills, you will live 10 years longer if you have hypertension, Now, do we believe that blood pressure pills affect omics data that we mentioned? Do you think blood pressure pills affect transcriptomics, proteomics, methylome, Maybe not, probably not, And so what I'm saying is the biomarker has to be adapted to the specific treatment, Having said this, what all of us want is a biomarker that captures a lot of the territory, right? And especially if you have an intervention where you don't quite know what to measure, right? But with blood pressure, it's obvious. Yes, blood pressure medication, of course, you measure blood pressure. If you have an HIV intervention, you measure CD4 T cell count. So often it's obvious what the biomarker is, but in some cases, it's not obvious what to measure. So for example, metformin, what should you measure, beyond glucose levels, or rapamycin, same question. And so then you have to hedge your bets, and like everybody else here on this panel, we are all serious scientists. We always want more data in order to maximize our chances to detect something significant, And I'll stop with that.
Peter?
Yes, well, I of course appreciate what has been said here, that aging is complex and so on. But I think Vadim's question was on the practical side. And when you're doing practical things, sometimes bad decisions now are better than more sophisticated solutions in the future. So I think in many ways, aging is much simpler in mice. I would say that you have a practical issue. If you want to detect an effect of drug in an experiment in mice, you take any quantity which correlates to the remaining lifespan, which is the risk of death. You measure it two weeks after the treatment, and I guarantee you that 2 weeks after the treatment, all the high-frequency effects disappear, and if there is an effect on all-cause mortality in the future, it is there or not. So what I'm saying is that if you want to predict a point in the future, death in the future, you have to wait a little bit after the experiment is finished, and if the effect is still there, most probably it will remain in this animal for a very long time. I'm not sure if I was understandable here, but if you want to predict an event a long time ahead, you have to be sure that the effect of the treatment sticks. And you can detect such the effect of such an intervention just by observing the animal long enough time after the experiment, after the intervention. If it's there, it will be longer or shorter, 60%. So that's my educated guess. Any risk factor 2 weeks after the treatment.
Yeah, thank you. Just before we continue with my questions, actually, there's a question from the audience, and I would like you to answer really, really short, in a short way. Just tell me just maybe one word or just one sentence. Yeah. So the question is, which biomarkers would you use for rapamycin human trial and also for metformin? like all of you, just tell the audience.
Maybe I'll start. For rapamycin, I would measure skin cells or buccal cells, anything related to keratinocytes. Why? Because in vitro studies show a beneficial effect of rapamycin in human keratinocytes.
But which biomarker specifically, not just which tissue?
Oh, epigenetic clocks. Thank you. So we have an epigenetic clock called the skin and blood clock. It applies to skin cells. It works beautifully in keratinocytes. The other biomarker is my original pan tissue clock. I would have again applied to buccal swabs, so that's what I would use for that. When it comes to metformin, I would use the GrimAge clock. It's a blood-based clock. We do not yet know whether metformin reverses the epigenetic age of blood. The jury is out, but that's what I would use.
Okay, thank you. Somebody else?
Well, we have with Andrei Gutkov, we tried rapamycin in mice, and we see that biological clock, risk clock, so the clock like phenol age or GrimAge, so the clock which is trained to predict mortality in mice, using blood cell counts and biochemistry, like PhenoAge works in mice. So I would try PhenoAge in humans in rapamycin study, but of course I don't know the answer.
I will track insulin resistance, because one of the side effects of the rapamycin treatment is increasing insulin resistance, so insulin and glucose. And on the other hand, I will track some proxy mTOR markers like insulin-like growth factor 1, which will definitely go down during this treatment, but shouldn't go down too much. So I will track these two groups of markers.
Thank you. Kristen?
Yeah, so you definitely want those markers just to see that the rapamycin was having an effect at all. But then you'd also want to layer on top some kind of omics marker to see that it was actually relevant for longevity. So my answer is basically the same as for our trial. You would do something like a methylome or a proteome. You keep your options open. Didn't someone publish that in mice, at least, taking rapamycin reversed some of these clocks? Am I misremembering that? Was it one of you guys?
Yes, we have a preprint. It's not yet accepted.
Yeah.
Okay, yeah, thank you. Let me ask you a little bit more detailed question about the biomarkers and clocks. What do you think they actually measure? Can you give actually molecular basis and how that relates to the aging process in a simple way, just so that's understandable for everybody? Yeah, go ahead. Too tough question for everybody.
There's so many different things going wrong as you get older, and it's just one number that summarizes all of them, right? I think Peter touched on this earlier. There are some clocks that are just combining all this information and you don't quite know what the mechanism is. And if all you want is just a metric that's really good at predicting the future that reflects intervention, you might not be able to figure that out. Certainly you can, build more specific clocks around specific molecular hypotheses, but I think it's often a hard question.
Yeah. Okay.
Yeah, just to echo that, some biomarkers could be called integrators, right? They integrate different innate pathways of aging, and therefore they don't have a simple explanation. From the view of epigenetic clocks, I can tell you that some of our epigenetic clocks, for example, relate weakly to blood cell composition and immunosenescence, emphasis on weakly, but they capture it, Then other epigenetic clocks relate to hematopoietic stem cell biology, And then there are other clocks which are much harder to interpret, where we believe they measure epigenetic stability, epigenomic maintenance. So in certain ways, they measure to what extent cells maintain their cellular identity, because the epigenome very much plays a role in maintaining cellular identity, But I already mentioned now several pathways, from immunosenescence to cellular identity to stem cell biology, and you can see that it's complicated. And the reason is perhaps because aging is complicated, So these biomarkers are integrators. Then there are other beautiful biomarkers that have a very simple explanation. Telomere length. You know, everybody loves telomere length because it's so well understood. The unfortunate thing about telomere length is that it's such a dismal predictor of morbidity and mortality. You know, so that's the problem with that. So as a biomarker person, you have to weigh things. Do I want to use a biomarker that works well in epidemiological studies? But am I willing to give up on biologic interpretability? Or conversely, you have a beautiful biomarker that relates to cellular processes, but then it may fail in epidemiological studies. So it's a tough call.
Yeah. Okay, Peter, would you like to continue?
Well, once again, I don't think that everyone understands the answer to this question in entirety at this moment. I can try to give maybe an engineering, a plausible engineering explanation to that. I think nature didn't build a clock inside us to help us with clinical trials. So that was not the point. So what I think actually happens is that, well, all-cause mortality is organism level phenomenon. So I think that our body has molecular pathways which regulate whole organism response to stresses. So what actually these biomarkers like mortality markers measure in healthy people, that's important, is that these are just stress responses. So that's why biological age is higher in people who smoke, and that's why it goes back to normal once you see smoking. Well, when you have diseases, this is just another source of stress, and that's anecdotally, this stress age actually tracks the total number of diseases which you have developed. So in my view, we're measuring stress, the integrated stress, the whole body response to stress, which essentially by chance also measures the total number of diseases which we have developed, which of course correlates to our age and the median lifespan. That's the best understanding of this moment.
Stanislav, what are your thoughts on that?
I think that maybe I want to add to what Peter is saying. Maybe most of the clocks are looking at the aging and mortality as a, so to say, gradual process, which is developing in time in some long periods, and like is putting us, biologically speaking, in this or that age category. But sometimes there are two things. One is stress resistance, resilience of the organism, and another one is the exact stress we are incoming. And therefore, we would very much like biological clocks to predict mortality, to understand how much time is left, so to say. But stresses do not come in a gradual and dosed manner. They come sometimes unexpectedly, like a black swan, like in a fragile environment. They cause too much damage to us. So this may be bringing us to some limitation of the biological clock method. So they are measuring aging as a gradual process, but mortality is not gradual. It's discrete happening, and which leaves us with a limitation here.
Yeah, Peter, yeah, go ahead.
I'm sorry for jumping without proper turn. Well, I think what Stanislav is saying actually comes from the observations. So we have lots of longitudinal data on humans. And what we see is that any biological age you like actually oscillates in healthy humans. And the amplitude of the oscillations is also the biomarker of age. So the more stable is your organism, the faster your organism responses to stresses, the less is the range of the fluctuations of your biological age. So not only the mean number, which we access in epidemiological studies and use to train the models of chronological age, but also the range of the fluctuations is both the property of your lifestyle. I mean, stresses do not come, according to a schedule. So both the amplitude of your stress, the force, the power of your stress commands your mortality, but also the resilience, how fast your organism comes back from the perturbation also commands your mortality. So in the future, once we jump from epidemiological studies to longitudinal studies, we will see that not only the level of biological age is the marker, but also the resilience and the power of the noise, the stress, are also markers which you can never get from a single measurement. And these are all very important things.
Thank you. Yeah. Next, I'm going to ask a question from the audience. And the question is, what do you think of making a focused push to utilize easily collectible physiological biomarkers to do something useful as a way to win public and policy makers of the value of biomarkers more generally? Anybody would like to comment on that?
Yeah, I mean, I think what we want to do as a field is to engage with regulatory agencies to convince them that there has been huge progress behind the scenes on aging biomarkers. And we have now entered a new time period where we can rigorously test promising anti-aging interventions, and we can rigorously test and try to repurpose various treatments. So I think based on the revolutions that have taken place in terms of technology, from the Human Genome Project to the most exciting proteomics measurements, and also great advances in our understanding of the biology of aging. Now is the time to invest in that, to build also the regulatory structure so that people can form companies, that exploit these basic science innovations. And so I think that it's very important to have an advocacy group that works on that.
Thank you. Yeah, Peter, go ahead.
I think that it will be very important to actually help people explain what are these biomarkers of age, how are they related to their lifestyles, how they are reflecting their mental health, for example, stresses and so on. I think the roadblock here is that the invasiveness of the procedures. I think that all of us would do blood work once for fun, maybe 2 times, but when you want to do a trajectory, when you want to see what's going on, I think few people will do it many times if they are not sick. So I envision that maybe within a few years from now, there will be maybe a company which would be using some progress in wearable devices, most obviously, so that people could actually give their information on their biological signals digitally without actually going to the doctor. And then this kind of people and this kind of companies will eventually test all pharmacology which is prescribable, and all kinds of gym sessions, all kinds of interventions, and actually score them, see what works, what not, and maybe even personalize it to every one of us. I think that would be a cool proposition if it ever made work.
Yeah, thank you. Kristen, would you like to continue?
Sure. So yeah, I would like in the future for there to be a biomarker of aging that we believed in enough such that it could be the primary endpoint in a clinical trial, like an approvable endpoint. I do think that future is still far away. I think the major barrier, the needed thing, of course, is to show that it works, right? So today, any clinical trial you're doing, it can certainly be a secondary endpoint, an exploratory endpoint. And then over time, you can show that it predicts these longer term outcomes that people really care about, like longer term mortality, hospitalizations, like hard functional outcomes that people associate with aging. So I would like to see, more exploratory analyses done in the range of clinical trials, because there certainly are drugs now that should be affecting the aging process. And that's the kind of evidence base we need to really build to get these approved.
Mm-hmm. Yeah, thank you. And Stanislav?
I definitely
briefly jump in. Yeah, I have a little bit a more charitable view of the field, from Kristen. So my analogy is always iPhone version 1 versus iPhone version 10, right? I think when it comes to biomarkers, we are at the level of iPhone version 1. We do have wonderful biomarkers that can already be deployed in human clinical trials. Yes, 5 years down the road, we will have much better versions, but the field is ready to build, to use these biomarkers. The other thing I want to emphasize, it's always plural. There's not one biomarker. We need to discuss what handful of biomarkers should be measured, or make it 10, All of us agree, measure CRP, albumin, all of these clinical markers. I think I want to say there is consensus that most clinical trials would want to measure methylation unless you have a good reason not to measure it, you know. But the point is, I think we're at the stage where we have already these biomarkers available, and we shouldn't wait. Why shouldn't we wait? Because we are aging at an exponential rate. We need to start these clinical trials now.
I want to cut in, Steve, because we actually weren't disagreeing. so definitely we can measure biomarkers today, and we're going to do that next year in our clinical trial. But what we can't have is an aging biomarker that's the primary endpoint, where that's the thing the FDA looks at and sees it move and says, yes, your drug is approved. The way that, you know, if you move cholesterol, right, you can get a drug approved for that. So that's a biomarker that's an approvable endpoint. So right now you can certainly measure biomarkers, but you still have to see, you know, a functional change or a disease change in order to get the drug approved. And I think it's still going to be a while before that changes.
Yeah. Okay, Stanislav, could you please continue?
I would just absolutely support any idea of an organized push for more testing, more data, more flow. That's just we need tools for this. I think that we want tools available for people to collect data and to process them, which will facilitate that. That's more or less what we are working on in this field. So I totally agree that there is a need for an organized push, not just some people speaking up here and there, but some maybe general strategy.
Okay, so we have just a few minutes left in the panel discussion. So I would like to actually ask you to express your opinion on what you think is the most important, what you would like to say, something maybe which we have not covered, but you think it's important to deliver to the audience. Okay, just kind of final statement, everybody. We covered everything? No.
No. Well, maybe I feel bad because I'm always the first to answer, but let me try. There's really wonderful news. Big picture is wonderful news. We have biomarkers that can be used in anti-aging clinical trials, very promising biomarkers, credible biomarkers. I also think expert panels can arrive at consensus views. All of us on the panel are quantitative people. So what I love about our field is we are all quantitative. We are all data-driven people, you know. We won't have big philosophical discussions. We will look at p-values and test, retest variability and replication, you know. So in that sense, our community is blessed, you know, that we will arrive at consensus. I think the field is ready, you know, to evaluate hundreds of very promising interventions. I think I could point people to various papers that discuss biomarkers. I'm happy to do it. I would mention markers of immunosenescence, flow cytometry. We mentioned all these traditional biomarkers of organ dysfunction, liver function tests, creatinine for kidney, and so on. And you know what other biomarkers I recommend. BioAge, I would strongly recommend. Yeah.
Thank you.
Yeah, so I'm also, you know, very optimistic about where we're going. Like there's a ton of clocks now, but look at how far we've come in the last decade, right? Like Steve, when was your paper first published? That was less than 10 years ago, right? And now there's just an amazing transformation in the field and an understanding of how these clocks are really important for different biologies and different data modalities, be it proteomics or metabolomics or methylomics. So we're right in the middle right now of figuring out what's important. And if we make progress in the next few years, like we have in the past decade, I think we'll get to consensus soon.
Peter or Stanislav?
I also think that nothing can be in the place of practice. I think everyone agrees that we have something to play with. So the more we are playing, both in animals and humans, the more we will understand, and I think we will learn a lot. So, really happy.
I want to add that maybe in the general, for people who are not engaged in science laboratories, the best option now, the best strategy is to invest in your own data, to collect it, And very soon, the methods, the tools, the platforms, the legislation will catch up. So, and when this all becomes available, then you will have your own data dating back a few years ago. That will be a very valuable asset for you.
Okay, thank you, everybody. I think we in 2 minutes, there's another session begins, so we probably need to finish now. There are still more questions from the audience, but I encourage the audience then to maybe just email the participants to get these questions. Just again, so our panelists were Steve Horvath, a professor from University of California, Los Angeles, a pioneer in developing the clock, who developed the first clock, I think, which is widely recognized now. Kristen, which has exciting new company that is actively pursuing this direction. And Peter Fedichev is also CEO of another company, which has also made great strides in the quantitative treatment of aging. And Stanislav Skakun is the CEO of BioData platform, which measures many biomarkers and also just very exciting direction. So thank you, everybody. I think it was very useful for the audience, and hopefully we can meet again next year and present our better updated view on biomarkers and clocks. Thank you again.
Thank you.
Yeah, thank you.
Thanks, everyone. Thank you, Vadim.